Life is a dance. It's a symphony of movements, a choreography of moments that blend together to create the beautiful rhythm of existence. It's a continuous cycle of growth, division, and renewal. Just as a dancer moves through a series of steps, life progresses through stages of development and transformation. At the heart of this dance is the cell cycle, a tightly regulated process that governs the life and times of every cell in our bodies. This cycle is the foundation upon which all life is built, ensuring that each cell performs its role with precision and timing. From the moment we are just a single cell to our adulthood, cells are constantly dividing, replacing old or damaged cells, and ensuring the smooth functioning of our tissues and organs. This relentless activity is what keeps us alive, healthy, and capable of growth and repair. Imagine a bustling city where old buildings are torn down and new ones are erected in their place. This urban renewal is akin to the cellular processes happening within us, where old cells are replaced by new ones to maintain the integrity and functionality of our bodies. This constant cycle of construction and renewal is what keeps the city alive and thriving. Similarly, our bodies rely on the continuous cycle of cell division and renewal to stay vibrant and functional. Similarly, the cell cycle is the engine that drives the growth, repair, and maintenance of all living organisms. Without this cycle, life as we know it would cease to exist, as cells would be unable to replicate and repair themselves. This incredible journey begins with a single cell. From this humble beginning, a complex organism emerges, built from countless cycles of cell division and differentiation. This cell must divide and create exact copies of itself. Each division is a precise and controlled process, ensuring that the genetic material is accurately replicated and distributed to the daughter cells. This process of duplication and division is the essence of the cell cycle. It is a fundamental aspect of life, driving the growth and development of all living organisms. It's a carefully orchestrated ballet with several distinct phases, each with its own unique set of steps and players. These phases include interphase, where the cell prepares for division, and mitosis, where the actual division occurs. Understanding the cell cycle is like appreciating a complex dance form. Each phase must be executed with precision and timing, much like a dancer must follow the choreography to create a seamless performance. We must first learn about the individual steps, the pauses, and the transitions. By understanding these elements, we can gain a deeper appreciation for the complexity and beauty of the cell cycle. Then we can marvel at the elegance and precision of the entire performance. The cell cycle is a testament to the intricate and delicate balance that sustains life. So, let's delve into the fascinating world of the cell cycle and explore the intricate steps of this dance of life. By understanding this process, we can better appreciate the complexity and wonder of the living world around us. Our first stop in the cell cycle is a stage known as the G1 phase. This phase is the initial part of the interphase where the cell spends most of its life. It's a period of intense preparation and activity, setting the stage for the cell's future endeavors. G1 stands for gap 1, but don't let the name fool you. The term gap might suggest a period of inactivity, but in reality, it's quite the opposite. This phase is anything but a gap in activity. The cell is bustling with energy, engaging in numerous processes that are vital for its survival and function. In fact, it's a time of tremendous growth and metabolic activity for the cell. The cell is actively taking in nutrients, converting them into energy, and using that energy to fuel its growth. Imagine a newborn baby rapidly growing and developing, learning new skills, and exploring the world around it. Just like a baby, the cell is in a phase of rapid development, constantly adapting and preparing for the future. The G1 phase is a period of intense biosynthesis. This means the cell is busy creating the building blocks it needs to function and grow. The cell is hard at work, producing new proteins, lipids, and organelles. These components are essential for the cell's structure and function. It's like a factory running at full capacity, churning out all the essential components needed for the cell to function and grow. Every part of the cell is involved in this process, working together to ensure the cell is ready for the next phase. The cell increases in size and volume during this phase, preparing for the next stage of the cycle. This growth is crucial because it ensures the cell has enough resources to support its functions in future division. This bustling activity is crucial because the G1 phase is also a time for the cell to carry out its designated tasks. Each cell has a specific role to play in the body, and it must perform these tasks efficiently. Just like each of us has a role to play in society, each cell has a specific function in the body. These functions are diverse and essential for the organism's overall health and well-being. Muscle cells contract to allow movement, nerve cells transmit signals, and gland cells secrete hormones. Each type of cell has a unique and vital role to play, and gland cells secrete hormones. These hormones regulate various bodily functions, ensuring the organism operates smoothly. All these tasks are carried out with gusto during the G1 phase. The cell is like a well-oiled machine, performing its functions with precision and efficiency. Think of it as the cell's day job. Just as we go to work and perform our duties, the cell is busy fulfilling its responsibilities. Before it can even think about dividing, the cell has to make sure it's fulfilling its duties and contributing to the overall well-being of the organism. This preparation is essential for the cell's success in the later stages of the cycle. The G1 phase is a critical checkpoint in the cell cycle, ensuring that only healthy and functional cells are allowed to proceed to division. This checkpoint is like a quality control system, making sure everything is in order before moving forward. It's a time for growth, work, and preparation for the next exciting stage of the cycle. The cell is gearing up for the S phase, where it will replicate its DNA and prepare for division. The G1 phase sets the foundation for all these future activities, making it a crucial part of the cell cycle. As the cell diligently performs its tasks in the G1 phase, a crucial question arises. How will it ensure that its daughter cells inherit all the genetic information they need to thrive? The G1 phase is a period of growth and preparation, where the cell increases in size and synthesizes various proteins and organelles. But the real magic happens in the next phase. The answer lies in the S phase, the next stage in our cellular ballet. This phase is a pivotal moment in the cell cycle, where the cell commits to duplicating its genetic material. Without this crucial step, the cell would be unable to pass on its genetic legacy to its progeny. S stands for synthesis, and for a good reason. This phase is all about replicating the cell's DNA, the blueprint of life. DNA, or deoxyribonucleic acid, contains the instructions needed for the development, functioning, growth, and reproduction of all known living organisms and many viruses. Imagine a meticulous scribe carefully copying a precious manuscript, ensuring that every word and punctuation mark is replicated with absolute accuracy. This analogy helps us understand the precision required during DNA replication. Just as a single error in a manuscript can change its meaning, a mistake in DNA replication can lead to mutations, which might be harmful. That's essentially what happens during the S phase. The cell meticulously duplicates its entire genome, creating two identical copies of its DNA. This process is not only about copying the DNA but also about ensuring that the copies are error-free. The cell employs a variety of proofreading mechanisms to correct any mistakes that occur during replication. This duplication process ensures that each daughter cell receives a complete set of chromosomes, the thread-like structures that carry our genes. Chromosomes are made up of DNA tightly coiled many times around proteins called histones that support its structure. The S phase is a remarkable feat of molecular choreography. Enzymes unwind the DNA double helix, separating the two strands like unzipping a zipper. This unwinding is facilitated by an enzyme called helicase, which breaks the hydrogen bonds between the base pairs of the DNA strands. Each strand then serves as a template for the synthesis of a new complementary strand. This is where DNA polymerase comes into play. This enzyme adds nucleotides to the growing DNA strand, matching each base with its complementary partner, adenine with thymine and cytosine with guanine. The process is incredibly accurate, thanks to the remarkable fidelity of the enzymes involved. DNA polymerase has a proofreading ability. It can remove incorrectly paired nucleotides and replace them with the correct ones, ensuring the integrity of the genetic information. By the end of the S phase, the cell has doubled its genetic content. It's like a library that has made a perfect copy of every book on its shelves. This doubling is crucial because it prepares the cell for the next stages of the cell cycle, where it will eventually divide into two daughter cells. This duplication is essential for cell division because it guarantees that each daughter cell receives a complete set of genetic instructions. Without this precise duplication, the daughter cells would not have the necessary information to function properly, leading to potential cellular dysfunction or disease. The S phase is a testament to the elegant precision of life's processes, ensuring the inheritance of genetic information from one generation of cells to the next. This phase highlights the incredible complexity and efficiency of cellular mechanisms, reminding us of the intricate dance of life that occurs within every living organism. With its DNA successfully replicated, the cell enters the G2 phase, the final act before the grand spectacle of cell division. G2 stands for gap 2, but like the G1 phase, it's a hive of activity. The cell continues to grow, synthesize proteins, and produce organelles at an accelerated pace. It's like the final preparations before a major event, ensuring that everything is in place for a smooth and successful performance. During G2, the cell checks its work. It meticulously proofreads the newly synthesized DNA, correcting any errors that might have crept in during replication. This quality control step is crucial for maintaining the integrity of the genetic code. The cell also uses this time to manufacture the proteins and structures needed for cell division, such as microtubules, which will play a crucial role in separating the chromosomes. Think of it as a dress rehearsal before the main event. The cell is making sure that its costumes are ready, the stage is set, and the actors know their cues. By the end of G2, the cell is at its maximum size, brimming with duplicated DNA, organelles, and the molecular machinery required for division. The stage is set for the dramatic events of mitosis, where the cell will split into two identical daughter cells.